Good morning. Good morning, everyone. Good to see you. Happy Monday. Happy rainy Monday. Such a rainy summer we're having. Of course, I won't complain too much because we're getting rain. And many parts of this country are in severe drought right now. Good morning, everybody. I hope you had a nice weekend. Yes, please go ahead and sign into chat. So as people are coming in, I'll remind everybody that you have a lecture exam to take today, lecture exam 4. That needs to be submitted by 8 o'clock tonight. John, I'm so sorry you're having to deal with those Canvas problems.
It's so random. I really have no idea like why that's happening. Like I don't know. You know how your computer can like pre-put in your username and password? I've been doing it that way for months.
Yeah.
And now I can't log on, and like I called Canvas and they couldn't help me, so they gave me a phone number that was out of service, and I found a different phone number and left a message.
Just so you know, there are a lot of I don't know what you'd call them, frequently asked question pages on the website for Canvas. So Canvas belongs to a company called InStructure. And if you go to their website, they do have a lot of pages of, frequently asked questions and answers. And I know for sure that there's a page called I'm having trouble logging in. And I actually sent you that link in an email or in a message through Canvas, which I don't know. Now I'm thinking about it. You probably can't get. Although I think it goes to your email too.
So I can't like through email, which I can only access my school email on my phone right now because my phone's automatically logged in. It has me like I have to view the thing through Canvas. However, I think you actually messaged Lacey back since I messaged you through her through Canvas. So I got that. I'm going to look at that and hopefully fix it as soon as possible.
Yeah. So in other words, you don't have to go into Canvas to access these help guides. You can go to their website on any browser and you can get to these pages that have a lot of frequently asked questions. And that may help. And you may find additional contact numbers on that page. But yeah, there's something very unusual going on because it's not like you were never able to get into Canvas. Yeah, so there's something very unusual going on. And keep me up to date on it. We'll make sure we get you where you need to go.
Thank you. You got it.
All right, everybody. So you have your lecture exam 4 today. I will say, just as a reminder, you can choose when to take it today. So take a couple minutes and go to your lecture quiz that was due Friday night, the transcription and translation quiz. And if you didn't get as good a grade on that one as you would have liked, be sure that you read through my comments and adjust your notes if you need to. If there were some things maybe you weren't quite understanding, my comments may help. So be sure to adjust your notes so your notes are all set to go before you sit down to take the lecture exam, sorry. There was a couple of questions that people missed the most. One of them was a question that had to do with you choosing the correct type of RNA, mRNA, tRNA, rRNA. There were 4 questions, A, B, C, D. You had to tell me what type of RNA was being discussed. Remember, there's RNA inside of the ribosome, isn't there? There's RNA in there. It's called ribosomal RNA, rRNA. It's found in the large subunit and the small subunit. There's also protein in both of those subunits. But the rRNA is the enzymatic part of a ribosome. It's capable of performing a metabolic reaction, and that is building a peptide bond between two amino acids. So rRNA that sits in the ribosome is catalytic. It's able to help a chemical reaction occur. And that chemical reaction is the building of peptide bonds between amino acids. Now, there was another part of that same question that asked where in the ribosome are amino acids joined to one another, taken off the tRNA and added onto the chain of amino acids. That's in the P site. And a trick that I use to remember that is that P stands for peptide. It's in the P site that the rRNA in the ribosome attaches the incoming amino acid to the growing chain of amino acids, or the polypeptide. The tRNA comes into the A site, carrying an amino acid. Then when the ribosome translocates, that means moves from one codon to the next. When it translocates, that tRNA now shifts into the P site. And the rRNA is going to take that amino acid and attach it to the growing chain of amino acids with peptide bonds. The next time the ribosome shifts, the empty tRNA now is going to be in the E site, the exit site, and then it's going to leave. So
I think that question was a little bit confusing because if you look at the lecture, the start codon is in the P site. So it makes it seem like it starts there. So it took a little bit of like rewatching it to like understand that, like, yes, it starts at the P site, but every other one coming in goes to the A site.
That's right. And remember, just like transcription, there are 3 stages to translation. There's an initiation stage, and that involves that methionine coming in, matching to the start codon on the mRNA, and coming into the P site. But that's the only one that does that. Once we get into elongation, we start to see the site to site to site to site. The what am I trying to say? The manufacturing of protein. And these tRNA come in one after another, because every time the ribosome translocates, we now have an exposed codon underneath the A site. And whatever tRNA can match and bind to that codon is going to come in. Okay? Yes, it can be very confusing. And that's why that particular lecture material really needs some time and careful review.
It made sense why you said that, because there was lots of times where you'd have to like go back and rewind and listen to it again.
Yes.
And they're like, okay, maybe I didn't quite get that. And they're like, the third time, a light bulb.
In some ways, it's helpful to watch a recorded lecture because you can do that. But in other ways, it's better to be in a classroom because you can raise your hand and you can say, hold on, I didn't get that, you know, and we can do it again together. So, but at least you have the ability to rewatch if something doesn't hit. And again, very complex procedures, these transcription procedures and translation procedures, full of detail, lots and lots of detail, and lots of terminology.
I tend to operate in analogies. So like if I can think of an analogy, like especially a funny one,
Yes.
to like make my brain understand it, that's usually how I do it.
Anything that helps.
Yeah.
Anything that helps is worthwhile. Yeah. All right. So I'm going to go ahead and pull up our schedule for just a minute before we get into our lecture, our lab for today, which is about protein catabolism. I just want to show you what we have left ahead of us as the course rapidly completes. So give me just a sec, bear with me. If I can manage this, there we go. Hold on, let me try again. All right, you, get out of my way. Hold on just a sec. That's better. One more time. Okay. Now you should be seeing our schedule from our syllabus. So we're right here, Monday the 19th. Lecture exam 4 is taking place today. We're going to talk about protein catabolism this morning. This is the week, remember, that you have the immune system on your lecture topic list. It's the only lecture for the week because it's a long one. And again, for some of you, this may be the first time that you've had a chance to learn about some of the basics of how the immune system works. So one lecture topic for the week, but it's a hard one. Give it the time that it needs. On Wednesday in lab, we're going to have respiration. Specifically, we'll be talking about anaerobic cellular respiration. That's on Wednesday. And that is the last lab where we'll be talking about biochemical testing. We will have finished all of the different types of testing that we cover anyway in this course. So I want you to look ahead to the following week, which is the last week of the course before the final exam. You can see that the lecture topics between today's lecture exam and the final exam are very different from what we've been talking about so far. We have left talking about the microbes to talking about how we live and deal with microbes. Again, we're talking about the immune system first, a little bit about vaccination, a little on epidemiology, and we finish out with pathogens and how they spread. So this last series of lectures is quite unlike the rest of the course in that we're leaving behind learning about the function and structure of microbes and instead talking much more about how humans interact with them, particularly when they are pathogenic. Now, when it comes to laboratory, next week on Monday, I'm going to be talking with you about your very last laboratory assignment, which is an exercise in identifying an unknown microbe. You're going to receive some data, some test results, and it's going to be your job to use some tools, again, that we'll talk about next week. Use some tools to help you interpret the results and figure out what microbe you have, what bacterial genus and species you have. So you'll have a chance next week to finish out laboratory, essentially, by performing the kind of work that is done every day in diagnostic labs, microbiology diagnostic labs. You'll have an opportunity to practice what we've been learning by using test results to identify a bacterium. The last thing you'll be doing on Wednesday next week is your last lab practical. That will, of course, cover what we've gone through since the last lab practical. So carbohydrate, fermentation, protein, respiration. And then of course we have the final the first week of August, and we'll talk more about the final as it gets closer. But yeah, we are that close, folks, to the end of the course. So this week, we'll be finishing up learning about some of the more commonly used biochemical tests in the microbiology lab. These tests that we use to, again, help us identify microbes that arrive in the laboratory, either in patient samples or in environmental samples. All right. Any other questions? Any questions about the lecture exam today? Any questions about what's coming up this week? If they occur to you as I'm talking this morning, go ahead and type them into chat and we'll get to them, okay? There are some things on our questions discussion board for you to take a look at if you haven't already. There are some practice problems for you to take a look at if you haven't. All right. Okay, so I'm going to go ahead and pull up our slides for today. And that's what you should be seeing on your screen, the title slide, Protein and Amino Acid Catabolism. So when we were together last week, we talked about carbohydrate catabolism. We talked about pathways that microbes use to break down or catabolize carbohydrates. We said that all cells use carbohydrate for energy. This week, we're doing something different. This week, we're switching over to the breakdown of protein and what exactly cells use protein for. And this applies today for us to microbial cells, but this can also be translated to other types of cells as well. So we'll talk mostly about the breakdown of protein, but we will also talk a little bit about the breakdown or catabolism of amino acids. Remember, amino acids are the building blocks of protein. And microbial cells, like all cells, do have the ability to break down amino acids if they need to. So we'll talk about both things and which tests we can use to see what a microbe is capable of in terms of this kind of metabolism. So our objectives today, first of all, we're just going to review a little terminology around protein because it's very easy to use these terms interchangeably when it's not really appropriate. I'm very guilty of this. So we'll have a review of the protein terminology. We're going to talk about the most common reason that bacteria break down protein. As I said a moment ago, we know that cells break down carbohydrate in order to make ATP, in order to get energy. We need to understand why cells, why microbial cells particularly, break down proteins. What's that about? We'll discuss that. And then we'll talk about two very common tests that we use. One is called the Gelatin Hydrolysis test, and the other one is called the Urease test. And these are both related to the catabolism of protein. For each one of these, we'll talk about what the media is made out of. We'll talk about how to perform the test and how to interpret the test. Now, the other thing that we're going to attempt to do today is to understand, again, why and how microbes might break down actual amino acids. So we're going to talk about two pathways, deamination and decarboxylation. We'll define those pathways, what we're talking about when we use these terms. And then we'll explain and interpret results for two specific tests of amino acid catabolism. One is called the phenylalanine deamination test. Of course, phenylalanine is one of the 20 amino acids. And then we'll talk about the cysteine desulfuration test, and cysteine is another amino acid. But let's start today by reviewing some of these definitions of terms that we use when we're discussing proteins. Remember, there's a difference between a peptide, a polypeptide, and a protein. And these terms, sometimes we interchange when we really shouldn't. And as I said, I'm very guilty of that. I tend to use the word protein when I shouldn't. A peptide, by definition, is simply a short chain of amino acids, again, that are connected to each other with a special kind of covalent bond. And that bond is called a peptide bond. So the key for a peptide is the word short. We're going to call that chain of amino acids a peptide if it contains tens to hundreds of amino acids. Now that may not seem short to you, but it is if you consider a polypeptide is a similar chain, a long chain of amino acids joined together with peptide bonds, but this time it contains hundreds to thousands of amino acids. Remember, most functional proteins fall into the thousands of amino acids category. Proteins are big. Proteins are very big molecules. So if it's in its primary structure or the chain structure, we're talking about a peptide or a polypeptide. If that polypeptide then does what nature wants it to do, which is fold up into a three-dimensional functional shape, now you've got a protein. So all a protein is a polypeptide that has been folded up through interactions, through weak interactions between parts of molecules into a functional three-dimensional molecule. Remember what brings that chain into a three-dimensional shape? Things like hydrogen bonds and van der Waals interactions. These are not covalent bonding actions. These are attractions. Every amino acid has an R group, a functional group, and the functional group has chemistry. It might be a polar functional group or a non-polar functional group. It might be a charged functional group. It might be positively charged or negatively charged. That's going to allow amino acids to either be attracted to each other or repulsed from each other in that long chain. That's going to help that big long chain fold. It's going to come together and fold. Hydrogen bonding is going to occur in certain regions. Van der Waals interactions are going to occur in certain regions. That whole thing is going to become three-dimensional and functional. Remember, every single protein has a unique shape. It is the shape of the protein that allows it to do its function. If the shape changes, the function changes. Okay? All right. So protein gets catabolized by microbial cells. Protein gets catabolized by all cells. Your cells and my cells do it as well. It can be broken down into those amino acids. In other words, the cell can set about to breaking those peptide bonds that connect the amino acids, that hold them together. So essentially, it's taking that protein and it's chopping it up back into the amino acids that made up the polypeptide. In fact, in bacteria, the most common reason for catabolizing protein is liberating those amino acids. Why? Well, because if it has more amino acids, it can use them to build new proteins. The most common reason that microbial cells catabolize protein is to get the amino acids out. Because, like all cells, microbial cells are constantly building new proteins, and they need amino acids to do that. So a handy way to get amino acids is to get them from other proteins. So remember, microbial cells, in fact, all cells, catabolize carbohydrate to make ATP. They catabolize protein for a completely different reason. And in bacteria, the most common reason is to get those amino acids out. It is also possible, though, for bacterial cells to break down a protein, get the amino acids out, and then break down the amino acids. So it's also possible for them to catabolize those amino acids and not just turn around and put them into a new protein. The reason microbial cells do this is because they need some carbon. All life on this planet is carbon-based. And carbon makes up what we call the skeleton of most molecules, at least the macromolecules. So microbial cells need a source of carbon in order to build other molecules. That's an anabolic reaction. And they need to get the carbon from some other carbon-containing molecule. And the one that they use is amino acids. So it's much more common for a bacterial cell to take a protein and break it into its amino acids and then stop, turn around and use those amino acids to build new protein. But if it needs a source of carbon, it can also take the amino acids and break those down so it can get some carbon for building other molecules. Now, there are some very particular reactions involved when we break down amino acids. Remember, all amino acids have the same fundamental structure, right? So the type of reaction needed is the same, regardless of which kind of amino acid you're talking about. Bacteria can break down or catabolize any of the amino acids to get that carbon using one of several different reactions. The first one is called deamination. Deamination, and this type of reaction removes the amino group from the amino acid. Deamin, here's your amino. Deamination. Now, when you take the amino group off of an amino acid, what's going to be left is an acid and ammonia. And a whole big family of enzymes is required to do deamination. Now, there's also a type of reaction called decarboxylation. And like the name suggests, this kind of a reaction removes the carboxyl group from an amino acid. And what you're going to be left with after decarboxylation is a base and some carbon dioxide. And again, a whole family of enzymes is going to do this reaction. There is another type of reaction that breaks apart certain amino acids, but this one's a little more specific. This one's called desulfuration. So desulfuration can only occur on the amino acids that have sulfur in them, as the name suggests. There are only a limited number of those 20 amino acids that contain sulfur. Okay, for example, cysteine has some sulfur in it, so does methionine. So what desulfuration does is it takes the sulfur out of the R group in sulfur-containing amino acids. And it's going to convert that sulfur into hydrogen sulfide, H2S. That's a important compound because we're going to look for this compound when we test for desulfuration. Now, this is a much more specific reaction. It's only applied to a couple of different amino acids. There's one enzyme that does this, not a big family of enzymes. And that one enzyme is called desulfhydrase, taking off of the sulfur and sulfur-containing amino acids, desulfhydrase. Now, if you remember when we talked about the starch hydrolysis test, when we were discussing carbohydrate catabolism, we said that the amylase enzyme that some microbes have gets secreted out of the cell and out into the environment. And that amylase enzyme is considered an exoenzyme because it gets secreted. When we talk about breaking down amino acids, this is all happening inside the cell. So those big families of enzymes that do deamination and the big families of enzymes that do decarboxylation and that desulfhydrase enzyme that does desulfuration, those are all endoenzymes because they live inside the microbial cell. So remember, there's a difference between an enzyme that a microbe is going to secrete out into the environment to do some work and an enzyme that's going to stay internally and do some metabolic work. When it comes to breaking down amino acids, it's all happening inside of the cell with endoenzymes. That's exactly right, Lacey. Function follows form. That little phrase is one of the most important phrases in biology. Form or structure is critical to how a molecule functions. And when it comes to protein, it's absolute. In order for a protein to do the job that it's intended to do, it has to have one particular structure. And if it doesn't have that structure, it can't do that job. So today we're going to look at both kinds of protein catabolism. We're going to first look at the breakdown of a protein into its amino acids. And then we'll look at the breakdown of amino acids. So the first two tests we're going to examine are tests that look for the ability of a microbe to break down certain proteins into amino acids. And remember, this is the most common use of protein. Metabolically speaking, this is the most common reason that a bacterial cell would break down or catabolize a protein. Now, the first test looks at the ability to catabolize a protein called gelatin. You've all heard of gelatin. You may have consumed gelatin. If you remember from anatomy, just as a little side note, gelatin is one of the components of connective tissue in animals. Very, very abundant protein in animals. And like all proteins, gelatin contains a wide variety of amino acids. So we can test for the ability to break down gelatin into its amino acids. And the enzyme that does that work is called gelatinase. That's an easy one, right? Gelatinase breaks down gelatin into its constituent amino acids. This gelatin hydrolysis test is a lot like the starch hydrolysis test because microbes secrete gelatinase out into the environment. Remember, proteins are really big. Proteins are big, much bigger than a lot of the other molecules that are inside of cells. It would be really, really hard for a microbial cell to take gelatin into itself intact. It'd be hard. So what it does instead is it secretes an enzyme outside of itself, and it breaks the gelatin down out in the environment. And then it takes in the amino acids. Very similar to what we saw with starch. Starch is a massive molecule. It would be hard for bacterial cells to bring starch in. So instead, they secrete amylase. They break the starch down outside of themselves, and then they bring in the glucose. So gelatinase is one of these exoenzymes. And some bacteria make it and some bacteria don't. If they have it and they detect gelatin in their environment, they will secrete the gelatinase that will break the gelatin down into all of its lovely amino acids. And then they will absorb those amino acids. It's okay, Stephanie. I'm glad you made it. All right. So we're going to look to see whether or not bacteria have gelatinase using the gelatin hydrolysis test. And the other test we're going to use, this one is a little bit, it's not a direct test of protein catabolism, but it's related to it. So we include it here in the lab. And that one's called the urease test. Now, what this test looks for is the ability of some bacteria to catabolize not protein, but a waste product of protein catabolism. And that waste product is called urea. You've probably heard of urea, or you may have heard of urea. Urea is a waste product. It's a waste product for you and I as well. It has to be released from the body as waste. Some bacteria contain an enzyme, again, an exoenzyme, called urease. And what urease is going to do is break down urea, that waste product, and release ammonia in the process. So we're going to test to see whether or not certain bacteria have the urease enzyme. Again, this is an exoenzyme. So let's say a bacterium finds itself in an environment where there's a lot of protein around it. Maybe gelatin. So the bacterium releases enzymes, exoenzymes, to break down that environmental protein into amino acids. And again, it can absorb those amino acids. Well, in the process of breaking down protein that way, urea gets made. And urea is waste. So a bacterium doesn't want to leave a lot of urea in its world. It doesn't want to leave a lot of urea in its environment because urea is toxic. It's a waste product. So while it's secreting enzymes to break down protein in its environment, it's also, if it's able to, it's going to secrete some urease because the urease will get rid of that urea. Okay. We humans also have to deal with urea because we humans also break down proteins. Our kidneys do this work for us. Our kidneys help get rid of urea. And those of you who have experience in the healthcare field, you may have heard of a test called blood urea nitrogen, or BUN. And a BUN blood test helps a physician know how well the kidneys are managing getting rid of urea. If they're not doing a good job at it, the urea is going to build up in the blood. And that's not good. It's a waste. It's a waste product. So we deal with urea too. It's just one of the products of breaking down protein, but it's not something that has nutritional value or anything like that. Okay, so the gelatin hydrolysis test is going to tell us whether or not the bacteria can secrete gelatinase, which is an exoenzyme. And the urease test is going to tell us whether or not the bacterium can secrete urease, which is also an exoenzyme. Let's start with the gelatin hydrolysis test. Now, this test uses a particular type of media called nutrient gelatin medium. And what this is a modified nutrient broth. So you start off with plain old nutrient broth, which is a basal medium, and you're gonna supplement it with gelatin. Look at the recipe for this medium. Boy, oh boy, what a short recipe. It has an enzymatic digest of gelatin in it. In other words, it has some gelatin that is partially digested. It has some intact gelatin, and it has something called beef extract. Beef extract. What do you suppose that is? Let me tell you a little bit about that word beef. Beef is a term that is used to describe a product that comes from cattle. The term beef does not specify skeletal muscle. It's just a tissue product that comes from cattle. So for example, when you pick up a package of hot dogs, not to be gross, but when you pick up a package of hot dogs and it says beef hot dogs on the package, that doesn't mean that those hot dogs are made out of the skeletal muscle components of that animal. It means that there's skeletal muscle in there, and there's connective tissue in there, and there's some blood vessels in there, and all kinds of stuff that was harvested from that carcass. So beef is a very complex kind of product. It's not just what we think of as the meat, the skeletal muscle. So if you put beef extract into something, that's a complex ingredient. I mean, that's got a lot of stuff in it. Yeah. Just so we know, not to put you off of hot dogs or anything, but when you see that word beef, it's a little bit non-specific. Okay. So we've got whole gelatin, we've got partially digested gelatin, and we've got this beef extract. All right, now here's where nutrient gelatin medium can be a little bit confusing. Nutrient gelatin medium is both a liquid and a solid, depending on what temperature it's at. Now, you could say the same thing for agar. Remember, agar does liquefy, but agar liquefies at a very high temperature. It's in the neighborhood of about 100 degrees centigrade. Nutrient gelatin medium is going to be liquid at or above 25 degrees centigrade. So it's a solid below 25 and it's a liquid above 25. It actually solidifies at room temperature. Room temperature, if you're not very familiar with Celsius, is about 22 Celsius. So at room temperature, this nutrient gelatin medium is going to be a solid. If you start to warm it up from 22, when you get to 25, it's going to liquefy. And then it's going to stay liquid above that. But at room temperature, it'll be solid for you. Okay? Elaine says, tasty talk about beef, especially in the morning. Right, Elaine? Oh, my goodness. The beef is put in there in case you forgot your snack. That's right. You can whip out some gelatin media, have a little beef snack at work. That's right. Okay. Now, why is this important to know about gelatin media? Well, when we inoculate gelatin media with bacteria, and that bacteria happens to be able to secrete gelatinase, the gelatin is going to get catabolized. It's going to get broken down. It's actually being hydrolyzed by the gelatinase. That word just means using water to break it down. And what that means is the gelatin's going to be gone. And in its place, there's just going to be a bunch of amino acids. And amino acids don't solidify at room temperature. So if the gelatinase is secreted and the gelatin gets broken down, that medium is now going to be a liquid at room temperature. Okay. So this is going to be important to us. We're going to read this tube at room temperature. If it's a solid, we know that there's gelatin still in there. If it's a liquid, we know that the gelatin is gone because the bacterium has broken it down. We do have a problem though, and it may not be immediately evident. Gelatin melts, becomes a liquid above 25 C. What temperature do we often incubate at? Anybody remember? Around what temperature centigrade, Celsius, do we incubate bacteria? Type it in the chat if you remember. It varies a little bit, but generally speaking, around what temperature do we incubate bacteria at? Anybody remember? Yeah, John's got it. 35 to 37. Yeah, Connie, warmer than that, right? Warmer than room temperature. Bacteria like it warm. So yeah, we typically set an incubator between 35 and 37 C. So we got a problem, don't we? Our gelatin medium, which is a solid at room temperature, so we can spread bacteria on it. If we put it in the incubator, it's going to melt. We can't incubate it in the incubator. We have to incubate it at room temperature. And we do that just by placing the inoculated tubes just on the countertop. We haven't seen a lot of this yet in here. We haven't seen a lot of room temperature incubation. You can incubate bacteria at room temperature. That's not a problem, but it's not the optimal temperature. We talked about conditions that affect the growth of bacteria, and one of them was the temperature of incubation, wasn't it? If you put a tube of bacteria on the counter, they will grow, but they're going to grow slowly because they're not at their optimal temperature. They're not at their optimal temperature for growth. You have to leave these tubes, these gelatin hydrolysis tubes, for about 5 days on the countertop. So it's a long incubation. And that's because we do it at room temp. Okay. Oh, actually, I wrote 7 days here. It's going to vary on the species of bacteria. So I'm going to just adjust this 5 to 7 days at room temp. Okay. Got to wait that long to make sure that the bacteria have had enough time to grow. And if they can secrete gelatinase, they will. Now, I do want you to notice a couple of things about the media before I leave this page. There's no agar in here. There's no agar in here. And yet, this is going to be a solid media at room temp. Remember, it's the gelatin in there that's going to make it solid. We purposefully don't put agar in here because the way we're going to read this test is whether or not the media stays solid. If the gelatin gets digested, in other words, if that bacterium can secrete gelatinase, the gelatin's going to go away and the media will become liquid. If there was agar in there, that wouldn't happen. Agar stays solid until a very high temperature. It's one of the reasons we use it. But the gelatin is going to be, it's going to give us a visual clue if it's been broken down. It's going to turn to liquid. And this is what it looks like. In this picture, you're looking at 2 test tubes that have gelatin, nutrient gelatin medium in them. This is what it looks like before you inoculate it at room temperature, okay? It looks like a deep tube, doesn't it? This is not a deep tube. This is not agar. This is a gelatin hydrolysis test tube. This is nutrient gelatin medium. You pour it into the test tube, you let it solidify at room temperature. Now, what we're going to do is we're going to put some cells in here, and then we're going to incubate for those 5 to 7 days, and we're going to look for this. Notice that the tube is being turned on its side here. That's important. You got to do that to read the test. This is turned into a liquid. It looks like a slant, doesn't it? It's not a slant. This is liquid in here now. So if you had this test tube in your hand, what you would do is tilt it from side to side and sort of roll it around, and you would be able to see that what was once solid medium has turned into liquid. The gelatin is gone in tube A. It has been digested away into amino acids. So if I were to take a little bit of this out and test it, I would find amino acids in there, but no gelatin. So you inoculate it, you put it on the counter, you leave it for 5 to 7 days, depending on the species. Then you pick the tube up. Imagine this is a test tube, and you do this. And you look to see whether that medium is still solid or if it's turned to liquid. If it's turned to liquid, you have a positive test result. That bacterium has digested away the gelatin, and the only way it can do that is if it has gelatinase, if it has the ability to make and secrete gelatinase out into its environment. If you pick that tube up after 7 days and you roll it around a little bit and the medium is still solid, that's a negative test. that bacterium does not have gelatinase. The gelatin is still outside in the environment. It's still solid at room temp. All right, let's take a look at the urease test. This is what the urease media looks like. It's got urea in it, obviously. It's got some sodium chloride. There's a little bit of salt. It's got some monopotassium phosphate, again, some minerals, some peptone, partially digested protein, some dextrose, some phenol red, that's an indicator dye, and some agar. So we know we're going to deal with a solid media for the urea test. Remember, it would be possible to take the agar out if for some reason I wanted to make this a broth. I could do that. But most of the time, we don't use a broth. We use solid media for this test. And we use it as a slant. Now, quick question. What is dextrose again? What's dextrose? What's dextrose another word for?
Sugar.
Sugar. What kind? It's simple. Good. Anybody remember what it is? Yes, Elaine, glucose. Glucose. That's what dextrose is. So we've got some partially digested protein in here, peptone. We've got some sugar in here, dextrose. Awesome. And we got a few little minerals for him. We've got sodium chloride, we've got potassium, we've got phosphate. Awesome. We have a nice little media here. And we have some phenol red. Now we've seen phenol red before. It's one of several indicator dyes that we've looked at. Phenol red starts off red. If an acid is produced, in other words, if the pH goes down, phenol red turns yellow. We've seen this before. One thing we haven't talked about, though, is what happens when alkaline conditions occur. What happens if phenol red is exposed to an alkaline condition where the pH is high? Wait till you see what color phenol red becomes. Again, urease medium is typically solid. You can make a broth if you want to, but most of the time we're using it as a solid and we're using it in a slant. Here's an interesting fact about this recipe. When you mix it up, it actually starts off slightly acidic. So when I mix this recipe up in the laboratory and put the phenol red in, the medium is not going to look red at the beginning. It's going to look yellow at the beginning because it's slightly acidic just to start with. So it starts off yellow. Remember, phenol red turns yellow anytime it's in acidic conditions, and it turns fuchsia in alkaline conditions. This is one of my very favorite colors in the microbiology lab, the urease, the positive urease test. It's a beautiful color, and you'll see it in just a minute. I'm going to take my urease test tube, that agar slant, and I'm going to spread bacteria across the surface of the slant with my inoculating loop. And then I'm going to put the tube into the incubator. I'm not going to leave it, though, for a full 24 hours. Instead, I'm going to check on it at intervals. I'm going to check on it every few hours. And that's because this reaction can occur quite quickly in some bacteria. If the bacteria that I have smeared onto the surface of that slant, if it secretes urease, what's going to happen is the urea in the medium is going to get broken down or catabolized, and ammonia is going to get produced. And ammonia is alkaline. That's going to turn the media a bright fuchsia color. And this is what it looks like. So over here on the left-hand side of the image, we have two auger slants. This one is what the media starts off as, what it looks like at the beginning. It has this yellowish color. Some people describe this as being a yellowish orange or even a peach color. But the idea is it starts off as more yellow than red. And that's because the media itself is a little bit acidic. I'm going to take some cells and I'm going to smear them on the slanted surface. I'm going to put them in the incubator. If this bacterium secretes urease, this is going to happen. Look at that color. You can't miss this result. This is positive. This means that the bacterium has secreted urease and has broken down the urea in this medium and produced ammonia and changed the pH to alkaline conditions. If the organism does not secrete urease, the media is going to stay this yellow color. This would be a negative test result. Now, like I said, you can make up a urease broth. The colors are going to be very similar. Here's what it looks like before you inoculate it. Here's what a positive test looks like. Here's what it would look like if it was a negative test and the urea had not been broken down. Now, I said that you don't want to leave this one in the incubator for the full 24 hours. You don't want to inoculate this test and just put it in and forget about it for 24 hours, because some bacteria secrete a lot of urease very quickly. And it turns out that's an important characteristic to know about a bacterium. There are some bacterium that secrete urease, but they don't secrete it very quickly in large amounts. And it will take them 24 hours or even more to turn the tube pink. But there are a few species that secrete the urea so quickly and in such large amounts that that tube is going to turn pink within about 6 hours, which is really fast in microbiology world, very fast. And like I said, you want to know that. If you're trying to identify this particular bacterium, you're going to want to know if it's rapidly urease positive. And by that, we mean within about 6 hours. If it can turn that tube pink within about 6 hours, we're going to call that rapidly positive. All right, let's introduce a little clinical application here. Take a look at these images. This one on the upper left-hand side, this is an EM image. What an awesome picture. I know, I'm a nerd. This is a scanning electron microscope image. And it's actually taken from the lining of the stomach in a human. So all these little dots that you see, those are those little microvilli that we see on the surface of the stomach and parts of the intestine. See these little things right here? That one right there. There's one right here. There's one right here. These are bacteria. This is a bacterium called Helicobacter pylori. This is what a drawing of the organism looks like. It has a very unusual shape, doesn't it? It's kind of not a rod, but it's not long enough to be a sporillum. It's kind of halfway between. Yeah, very unusual shape. And it's got a whole bunch of flagella at one end. Now, this image right here in the center, this is an image, again, taken from the lining of the stomach, but this is under a light microscope. So this is all stomach tissue. What you're looking at here, and I'll outline it, this is a gland. Remember, there are glands in the stomach. Some of them secrete mucus, some of them secrete acid. Glands have a cavity within them, if you remember your anatomy. And right here, right in the middle of that cavity is a bacterial cell. Helicobacter pylori. This is a smear that was stained. Hard to see again because it's a light microscope image. Look at the strange shape of these. You would be forgiven if you called these spirilla. They look like spirilla, but we don't tend to call them spirilla. We tend to call them elongated rods. They don't really meet the definition of a spirillum. They don't really meet the definition of a rod. Very unusual shape, an elongated rod. Now, why am I talking about Helicobacter? Helicobacter is an important pathogen in human medicine. And believe it or not, yeah, Lacey's got it. Believe it or not, we haven't known about Helicobacter all that long. When I was a child, if you were diagnosed with a gastric ulcer or a small intestinal ulcer, a duodenal ulcer, your doctor would have looked at you and said, it's stress. You're stressed. That's why you have an ulcer, because you're stressed. Well, some years ago now, a researcher discovered that there was actually a little bacterium in a lot of those ulcers. Not all of them, but enough of them so that now any person who is diagnosed with a gastric or a duodenal ulcer will be examined for this particular bacterium called Helicobacter pylori. I want you to just sit in awe and amazement at this little bacterium. It lives in the stomach, where there's a pH of about 1. It manages to get into the mucus that lines the stomach, and it gets into those glands where the mucus gets secreted, and it's protected from the acid. But it's pathogenic. It destroys tissue and it creates ulcers. So again, nowadays, if you get diagnosed with an ulcer, they're going to go and get samples, and they're going to figure out whether or not you have H. pylori infection, because it's a bacterium and it can be treated with appropriate antibiotic therapy. Now, it's important to know that not all ulcers are caused by this bacterium. There are some ulcers that are caused by other conditions in the body. But it turns out quite a few of them are caused by H. pylori. Now, where does microbiology come into this? Well, one of the cool things about this organism is that it is rapidly urease positive. One of its traits is that it is rapidly urease positive. So what will happen sometimes is a patient will come in to have a biopsy taken of their ulcer, and the surgeon will send that biopsy material straight to the microbiology lab in the hospital, and that biopsy material will be used to inoculate a urease test tube. And that tube will go into the incubator, and it will be checked every hour or so. And if it turns pink by 6 hours, that gets reported back to the physician, and a presumptive diagnosis is made. A presumptive diagnosis, of course, because it's only one test that we've done. But when you're dealing with an ulcer in a patient, a gastric ulcer or a duodenal ulcer, and you get a biopsy and you use that to inoculate a urease test, and it comes back rapidly urease positive, it's a pretty good bet that there's Helicobacter in there. So it's nice because the doctor and the patient get a very quick result there. That patient will be started on antibiotics. Now, we're not done in the microbiology lab. We're going to take that biopsy material and we're going to do other tests as well because we want to confirm the diagnosis. But yeah, if you get a rapidly positive urease, you've got a presumptive diagnosis, and that patient can immediately get started on antibiotics. So there is a very practical use for the urease test. Now, it's important to know that Helicobacter pylori is not the only organism that is rapidly urease positive. If we leave behind gastric ulcers for a minute, and I'm just going to enlarge this down here because I put some notes for you. There's another type of pathogenic microbe that a rapidly urease positive test result, it's very helpful in diagnosing. We've talked about fungi in here, and we've talked about yeast. Well, the most common pathogenic form of yeast, in humans anyway, is in the genus Candida. Here's the word down here, Candida. There's a very common type of Candida called Candida albicans. Pathogenic strains of that can grow at various parts of the body. There's another kind of Candida called Candida neoformans. So a couple different Candidas. Again, not all Candida is pathogenic. You're covered in Candida, okay? You've got it inside of you, you've got it outside of you. Very common part of your microbiome, Candida. But there are pathogenic strains. It can be difficult sometimes to differentiate those strains in the laboratory because when you grow Candida on an agar plate, for example, the different species tend to have similar colony morphology. So what can you do? Well, one of the things you can do is you can run a urease test because Candida albicans is going to be urease negative. Candida neoformans is going to be rapidly urease positive. So that's going to be some important information if you're ever trying to differentiate among those yeast. If you've got a pathogenic yeast, you're not sure yet what the species is, one of the tests that you will run is a urease test. And Candida neoformans will give you an important result, rapidly urease positive. All right, let's run a couple of these tests, shall we? We'll do the gelatin hydrolysis test first. We're going to look at Staphylococcus aureus and Staphylococcus epidermidis on the urease test today. These are both gram-positive cocci. We've seen these organisms before. Remember, Staphylococcus aureus is found on your skin, but also in your nose, in the back of your throat. There are non-pathogenic strains of this organism, and of course, there are very, very pathogenic strains of Staph aureus. Staph epidermidis is also found on the skin. So these two species of Staphylococcus can be easily confused for each other, and we need to do tests in the lab to tell them apart. One of the ways we can test to tell them apart is with the gelatin hydrolysis test. What special media can I use to tell Staph aureus apart from Staph epidermidis? What type of special media that is both selective and differential? What type of special media can I use to tell Staph aureus apart from Staph epidermidis? They can both grow on it, but there's going to be a difference between them. Anybody remember? Connie's got it. Mannitol salt. Remember mannitol salt? Not very many things can grow on mannitol salt. Staph tends to grow well if it's of those two species. We can tell them apart because Staph aureus can ferment mannitol. Staph epidermidis can't. So aureus will grow and turn the media yellow. Epidermidis will grow and leave the media pink. Connie's also asking about this term, this Bacter term. It does have meaning, but nothing practical, Connie. A lot of the names that we give, scientific names that we give to organisms, come from Latin. Some come from the Greek. That's where that Bacter comes from. I think it's Latin. And again, it's nothing practical to us in terms of what it would mean. All right. So we're gonna compare these two organisms with the gelatin hydrolysis test. We're gonna run a urease test on these three organisms: E. coli, our old friend, Proteus mirabilis, and one we saw last week, Klebsiella pneumoniae. All three of these are gram-negative rods. All three of these can be found in the gastrointestinal tract. Remember, E. coli is a coliform. It can ferment lactose. Proteus mirabilis, it actually lives in the colon specifically, but it can also be found out in our environment. The pathogenic strains of Proteus mirabilis are pretty bad. This is an organism that can cause infections in different parts of the body. It's a relatively common cause of urinary tract infection, of respiratory tract infection. It can cause infection in burn patients. It can cause sepsis. Remember, when we use that term, we're saying infection that has gone into the bloodstream. Sepsis is very dangerous, kills a lot of people. Klebsiella pneumoniae is a little bit different. It is found in the intestinal tract, but it can also be found on the skin. And again, pathogenic strains can be really problematic. They can grow in different parts of the body. And like the name suggests, they can cause a pneumonia in the respiratory tract. So, three organisms that we will compare on the urease test. Here are the results for our gelatin hydrolysis test. We took the nutrient gelatin medium and we inoculated 2 tubes, one with Staph aureus and one with Staph epidermidis. The tubes were placed on the countertop and left for 7 days at room temperature and then tilted for examination. Here's epidermidis. Here's aureus. Which one of these is able to produce and secrete gelatinase? Epidermidis or aureus? Who has secreted gelatinase there? Type your answer into chat for me. Which one of those organisms secreted gelatinase and broke that gelatin down into a whole lot of different amino acids? and then absorbed those amino acids. Good. Lacey's got it. Elaine's got it. Connie and Stephanie and Valerie. Good. Urease. How do I know? I know because at room temperature, nutrient gelatin medium is a solid. The gelatin inside that medium solidifies the medium. If there's no gelatin in there, because it's been digested away, that medium is going to be a liquid at room temperature. So epidermidis hasn't done anything with the gelatin, has it? Because the tube is still solid at room temp, but Urease has digested it away. Here are our urease tubes. This tube that's labeled A was used as a control. It was put into the incubator uninoculated for comparison purposes. This is what the media looks like in an agar slant before it gets inoculated. It has a slightly yellowy orange color, and that's because the media itself is a little bit acidic. There's phenol red in here, and phenol red will turn yellow in the presence of acid. This tube labeled B was inoculated with Proteus mirabilis, and I can tell you it was pulled out of the incubator at about 6 hours. This tube labeled C was inoculated with Klebsiella pneumoniae. Ooh, this is an odd one. Pink at the top where we spread the cells, yellow at the bottom. And this tube over here, this is E. coli, the tube labeled D. Now remember, we have this tube as a control. First question. Has anything happened in this E. coli tube? Has anything happened in that tube? Or did the E. coli just not do anything? Lacey's got it. Connie's got it. That tube's not the same color, is it? Remember, a control tube is your comparison tube. If nothing had happened in that tube, if we spread the E. coli on and we put it in the incubator and the E. coli did nothing metabolically, the media would have stayed the same color. It did not stay the same color, did it? Look at those tubes again. Here's your control. Here's your E. coli. This tube is more yellow than this tube is. This tube is more yellow. This tube is much more what we think about in color when we think about phenol red in an acidic environment. The E. coli is doing something. It sure is, but it's not secreting urease, is it? If it had secreted urease and it had broken down the urea, ammonia would have been produced. The pH would have become alkaline, and the tube would have turned fuchsia pink. That didn't happen. But it is doing something, isn't it? You know what it's doing? It's metabolizing. It's growing and metabolizing. There's dextrose in this medium. There's dextrose in here. There's glucose in here. You know what it's doing? It's metabolizing the glucose to make some ATP, and it's secreting an acid and turning that phenol red yellow. That's all. It's just growing. It's happily growing. It's just not breaking down the urea. Connie says, "Aw, little E. coli, our friend E. coli, is happily growing." Remember, E. coli is a very adaptive little organism, and it's very happy to grow in all different kinds of media. even if it can't do what we're testing for. Now look at the other two. Who is our rapidly urease positive? Which one of these bugs is rapidly urease positive? These tubes came out, well, E. coli came out at 24 hours, we're going to say. But this tube and this tube came out at 6 hours. Type into chat who our rapidly positive organism is. Is it Proteus mirabilis? That's B. Is it Proteus or Klebsiella? Yeah, it's Proteus. Proteus turned that media fuchsia pink within 6 hours. That's a fast test result. Think about what that means. That organism grew and secreted enough urease to break down enough urea and make enough ammonia to turn that entire agar tube pink in 6 hours. Yeah, rapidly urease positive. Now, what about this Klebsiella tube? Half pink, half yellow. What the heck? What the heck? It obviously has some urease in it, right? It has obviously secreted some urease because we got part of the tube pink. Why isn't the whole tube pink? That's right, Lacey. That's right, Elaine. This is a positive test, but this is not rapidly positive. Klebsiella is growing in there. It's also catabolizing the carbohydrate, isn't it? Like E. coli's doing. That's why the bottom of the tube is that yellow color. And it is slowly secreting some urease, and it's breaking the urea down too. In fact, if you put that tube back in the incubator and you took it out at about 24 hours, the whole tube will be pink. But at 6 hours, no, it's secreting it just more slowly. It's breaking down the urea more slowly. There's less ammonia in there. It just hasn't done it quite as quickly as that Proteus did. So that's a positive because you got the fuchsia, but it's not a rapidly positive. Okay? Takes more time. That's right, Connie. Takes more time. All right. So let's switch gears now. Let's leave the breakdown of gelatin. Let's leave the breakdown of that waste product called urea. And let's focus more on amino acid catabolism. Remember, the most common reason that a bacterium would catabolize a protein is to get at those amino acids. It will absorb the amino acids and it will use them to build new proteins. But sometimes a bacterium will break down a protein, get the amino acids out, and then catabolize the amino acids. And that's because amino acids can be used as a source of carbon for the synthesis of other molecules. Now on this slide, over on the right, you're looking at the basic structure of an amino acid. You've seen this diagram before in lecture. All amino acids have the same basic structure. They have a central carbon that has 4 things attached. They've got an amino group, that's the purple end. They've got a carboxyl group, that's this end. They've got a single hydrogen, and they've got an R group. Remember what R means in chemistry. It means there's a bunch of atoms over here connected to each other that have some kind of function in terms of this amino acid. It's the R group that makes one amino acid different from another. Some R groups are polar, some R groups are non-polar, some are positively charged, some are negatively charged. The R group makes each amino acid unique. Here's another drawing of the amino group. Sometimes we draw the R to mean the rest of the molecule. Here's the amino group. Here's a carboxyl group. Here's a carbonyl group. Here's a phosphate group. We've talked about those. Here's a hydroxyl group. There are lots and lots of functional groups in chemistry, and we mentioned the ones that tend to show up in living cells at the beginning of the course or towards the beginning of the course. There are certain functional groups that tend to show up more often. But yeah, it can be a whole variety of different things attached as the R group to an amino acid. There's even an R group that contains a little bit of sulfur. That's a sulfhydryl group. See that one over here? But there are other R groups that aren't even shown on this screen. Some of them can be quite big, quite complex. So it's the R group that determines the unique nature of each specific amino acid. And what we're going to test for is how or whether or not a bacterium has the enzymes necessary to take apart certain groups on the amino acid. Some bacteria are going to have the family of enzymes needed to just remove the amino group. That's called deamination. All amino acids have an amino group. Some bacteria have a family of enzymes that can remove this carboxyl group. All amino acids have a carboxyl group. And some bacteria have a particular enzyme called desulfhydrase. And desulfhydrase can remove sulfur. Now, amino acids do not all have a sulfur group, but some, in fact, there is no sulfur group, but some bacteria have sulfur in their R group. A limited number of bacteria have an R group, a limited number of amino acids have an R group that contains some sulfur. We can check to see whether or not bacteria have the enzyme needed to remove that sulfur and turn it into hydrogen sulfide. So deamination can be done on any amino acid. If the bacterium has the necessary enzymes, any amino acid can be deaminated. Any amino acid can be decarboxylated, but only sulfur-containing amino acids, only the ones who have sulfur in their R group, can be used for desulfuration. So there's a limited number of amino acids there. Two good examples are cysteine and methionine. Both of those have sulfur in their R group. We're going to do a test for cysteine. We're going to give bacteria cysteine, and we're going to see if they can desulfurate it using the desulfhydrase enzyme. If they have the enzyme, they'll do it. If they don't have the enzyme, they won't. All right. The first test we're going to do is for deamination. And this one specifically uses phenylalanine. I can't tell you why this test uses phenylalanine. I don't know of anything particularly special about phenylalanine, except that we know exactly what product molecules are produced when you deaminate phenylalanine. So for this test, we're going to use a plain old nutrient auger, and we're going to supplement it with phenylalanine. And we're going to put this agar into a slant. Now, when phenylalanine gets deaminated, two things are left behind: ammonia and a molecule called phenylpyruvic acid. Now, this test is a little more complex than a lot of the tests we've looked at, in that we can't just take the tube out of the incubator and read it. We have to add a reagent to it. Remember, we saw this with the VP test too. Remember, we talked about the MR VP tests that go together? And we said when you pull the VP tube out of the incubator, you got to put a reagent in. And you look for that red ring. Well, the phenylalanine deamination test also needs a reagent. And it's a reagent that contains iron. Okay, keep that in mind. Lacey's made a comment about the PKU test. Very good. Phenylketonuria. That's right. And this is a test that is done on all newborns. Yes, because if a newborn baby has phenylketonuria or PKU, it can kill them within a matter of months. That's right. And what we look for in PKU is a buildup of phenylalanine, right? Because some little babies don't have the ability to break down phenylalanine. And phenylalanine is a common amino acid. And what happens in those poor babies is that phenylalanine builds up as they consume protein. And unfortunately, it builds up in their nervous tissue, their nervous system. Yeah, very good, Lacey. Good job. Oh, Connie, that used to be her job to do those tests. Yes, and you have to stick the little baby. I know. You have to poke them and get the little blood. I know, Connie. It's terrible. I know. But a very important test to conduct. Because if you can identify a baby who has PKU, it can be managed. That baby doesn't have to get ill. That baby doesn't have to die. You just need that baby to consume a diet that doesn't have phenylalanine in it. Those babies go on to live long and happy and healthy lives. They just need a dietary restriction. They just need to keep phenylalanine out of their diet. Very good. Yeah, the trick is, warm the foot up, right? So you can get the blood and put them on mom's breast. That's right. Get them nursing and distract them a little bit from what you're about to do to their foot. That's exactly right. Okay. So when we pull a phenylalanine deamination tube out of the incubator, we're going to add an iron-containing reagent called ferric chloride. When you mix ferric chloride with one of those product molecules, phenylpyruvic acid, you're going to get a color change. And the color this time is going to be a very dark green color. Not a pretty color this time, but very noticeable. That's a positive test. If you put ferric chloride into the tube and the media turns that dark green color, that's telling you that there is phenylpyruvic acid in there. And the way it got there is because the microbe secreted the enzymes needed to deaminate the phenylalanine. Those enzymes take the amino group off of phenylalanine and leave behind phenylpyruvic acid and ammonia. If you get no color change on this test, you've got a negative test. The bacterium does not have the needed enzymes to deaminate phenylalanine. And this is what those tubes look like. Phenylalanine deamination slants have this really boring color to start with, this sort of beige color. You can just see that this was an auger slant. Right there is the slanted edge of the auger. And over here, it's a little bit harder to see, but it's right there. So this is a solid medium. We generally conduct this test using an auger slant. Now, when you pull the tube out of the incubator, after 24 to 48 hours, sometimes it takes a little while, you're going to add a reagent. That's what you're looking at here in this brown gold liquid. This liquid that has been added in is ferric chloride. So again, here's the edge of the slant. And then we just dripped in an ml or 2 of ferric chloride and we let it sit. Same thing over here. Can you see the ferric chloride, that goldish brown liquid sitting on this slant? Only over here, now the auger has turned dark green. This is a positive test. Whatever microbe is growing in here has the enzymes necessary to deaminate phenylalanine. I know that because when I added ferric chloride, it reacted with the phenylpyruvic acid and we got a green color change. This one over here, this is a negative, no color change. This media actually does not have an indicator dye in it. When you mix ferric chloride with phenylpyruvic acid, you get a precipitate in the media. That's just a little trivia for you. It's not actually an indicator dye here. This is an actual precipitate that forms. And it has that dark green color associated with it. Now, cysteine desulfuration looks for that desulfhydrase enzyme. Does the microbe make desulfhydrase? Because that's the enzyme that can remove sulfur from amino acids like cysteine. Now, this particular test, cysteine desulfuration, uses a type of media called SIM media. And I'll talk about that in just a moment. SIM media is not just used for this test. It's used for several different tests in the micro lab. And you can get it as a broth, or you can get it as a slant, or you can get it as a deep. It's a very commonly used media because you can run different tests with it. If the microbe has the desulfhydrase enzyme, that means it can pull sulfur out of the R group of cysteine. It can catabolize cysteine. You're going to get pyruvic acid made and hydrogen sulfide, H2S. So remember, the phenylalanine test makes phenylpyruvic acid, phenylalanine, phenylpyruvic acid. The cysteine test makes pyruvic acid. And of course, another name for pyruvic acid is pyruvate. This is the same pyruvate that we talk about when we talk about carbohydrate catabolism. But interestingly enough, it's not the pyruvic acid that we're going to be able to see in this test. It's the hydrogen sulfide. When you take this tube out of the incubator, you're going to add a reagent. It's another iron-containing reagent. But it's not ferric chloride this time. It's ferrous sulfate. It's another iron-containing reagent. What's going to happen when you mix the ferrous sulfate in is if there's hydrogen sulfide present, you're going to get a black precipitate called ferrous sulfide. It's going to turn black if it's positive. If you get no black color change, you've got a negative test result. So just a little bit of information about SIM media. This term SIM stands for something. It stands for sulfur reduction, indole production, and motility. So SIM. SIM. We can use this media for several different tests. What we're using it for is sulfur reduction. That's what we're doing by adding that iron compound to the H2S. What we're getting is that black precipitate, sulfur reduction. Look at the ingredients that are in SIM media. A pancreatic digest of casein, which is a milk protein. So again, partially digested casein or milk protein. Peptone, which is another partially digested protein. Ferrous ammonium sulfate, that's a source of iron, right? And sodium thiosulfate, that thio term means we've got sulfur in here. And it also has agar in it. This test media, this is what SIM media looks like when you buy it, essentially. This is what the recipe looks like. We can't use SIM media as it comes from the chemical supply house. We have to make our own if we're going to use it for sulfur reduction. Because remember, we're going to add the iron compound. So we got to get this out of here. And we don't want there to be a sulfur source in here. Why is it important to take the sulfur source out of the SIM media? Why, if we were going to test for the ability of a microbe to break down cysteine, why would I want to be sure to remove all other sources of sulfur? Let's think about that. I'm testing to see if the microbe can take the sulfur out of the cysteine and turn it into H2S. I can see that H2S if I add an iron compound. Why would I need to take all the other sulfur sources out? What do you think? What would happen if I left another sulfur source in that media and it turned black and the media turned black? Would I know that the cysteine had been desulfurated? Would I know that for sure? Something got desulfurated, but would I know it was cysteine? No, that's right. That's right, Lacey and Connie. That's right. I wouldn't know. I wouldn't know which sulfur source got desulfurated. I'm going to put cysteine into my SIM media. So I got to take all the other sulfur out. I got to take out the iron and I got to take out the sulfur. And then I can add in cysteine. Now what I've done is I've given that bacteria only one source of sulfur. And that's the sulfur that is found in the R group of cysteine. If that organism can take that sulfur out of the cysteine, I'm going to find H2S in there when I add my iron, and it's going to turn black. So I got to get my SIM media adjusted specifically for this test. I got to get the iron out because I don't want to put the iron in until the end. And I want to get the sulfur out because the only sulfur source I want in there is the cysteine, because I want to know if it can desulfurate cysteine. So remove the iron, remove the sulfur, add cysteine. Add plenty of cysteine. And now I'm ready to test. This is what the media looks like. If you put it into a test tube like this, you can do this as a broth. You remember, you can do it as a slant. You can do it as a deep. It all depends on the microbe that you're testing here. This happens to be a pair of cysteine desulfuration broth tubes. How do I know? Because the black went all the way through. So right at the top here, see this little bit of sort of beigeish yellow liquid? That's that iron containing reagent that I dripped into the tube when I took it out of the incubator. And the whole broth tube will turn black. It's black because the iron has reacted with hydrogen sulfide and caused it to precipitate as black. This test is negative. I dripped in my iron containing reagent. I got no precipitate forming. That's a negative tube. Over here, these are cysteine desulfuration tubes, but this one was done as a deep. Remember, we'll use a deep agar tube if we're working with a microbe that can't grow around oxygen. Or maybe it needs a lower level of oxygen. It doesn't like room air. It has too much oxygen in it. So we take our inoculating wire and we dip it into the culture, and then we stab the wire down into the agar. Look over here. This is a positive result using an agar deep. See the stab line? That's where I stabbed the cells down into the agar. I dripped in some of that iron containing reagent, and I can see black. The cells were growing along the stab line, and they were taking the sulfur out of the cysteine and creating hydrogen sulfide. And when I added the iron compound, it precipitated out the hydrogen sulfide as black. This one's negative. You can't even see the stab line in here, but there is one. This is a negative result. This is a positive result. Questions? I can test using the phenylalanine deamination test. I can test if a microbe has the enzymes needed to deaminate phenylalanine. If they can do that, one of the products is going to be phenylpyruvic acid. And after incubation, I'm going to add an iron-containing reagent. If there's any phenylpyruvic acid in that tube, it's going to react with the iron, and it's going to precipitate and be green, dark green. If the organism doesn't have the enzymes to take the amino group off of phenylalanine, I'm not going to get a color change. I can test to see if a bacterium can take the sulfur out of the R group of cysteine. If it can, if it has the desulfhydrase enzyme, it's going to create pyruvic acid. And when I add an iron-containing reagent, it's going to react with the pyruvic acid and cause it to precipitate. Any problem with that? Do this just every once in a while to make sure you're listening. Is it the pyruvic acid that's precipitating in that test? What is it that's precipitating out in the cysteine desulfuration test? What's reacting with the iron compound again? It's not the pyruvic acid. It's the other product. What is the other product? Good. Hydrogen sulfide, H2S. If the microbe has desulfhydrase, it's going to take the sulfur out of the cysteine. We're going to get pyruvic acid and H2S. When I add the iron in, the iron's going to react with the H2S and cause it to precipitate out. And it's going to look black. Good. Two different tests that are testing the ability of a microbe to catabolize a certain amino acid. There are other tests that we can run for other amino acids, but those two are quite common. We can test for the deamination of other amino acids. We can test for the desulfuration of, say, methionine. There are other tests as well, but these are two common ones. And they tell us something about the microbe's ability to catabolize certain amino acids. And it would do that if it needs a source of carbon. That's why they sometimes break apart protein. Most of the time they break apart protein to get amino acids. But sometimes they actually break apart those amino acids because they want the carbon. And the reason they want the carbon is because they need to build something else. And carbon is the skeleton of all of the macromolecules. Very good. Good job. All right, everybody. When I see you on Wednesday, we're going to talk about some tests we can run concerning respiration, and specifically anaerobic this time. But for today, remember, you've got a lecture exam to complete for me. Make sure your notes are in order before you sit down to take that test. All right? All right, everybody, I'm going to let you go. Have a good day. Stay dry. I'll see you next time.